And it's a real pleasure to introduce John Yates, despite he not needing any introduction. I could easily spend the rest of the day telling you about all the great things that John has done, and I would still not have enough time to enumerate everything. But I would go for short, and short is innovation. If I wanted to associate John with one word in my mind, the thing that first comes to me is innovation. He's been a key innovator in just about any important technology in the realm of generating and analyzing mass spectrometry data, starting from his seminal contributions to interpreting mass spectra and assigning sequence with sequest, and his contribution to the development of data-dependent acquisition methods, data-independent acquisition methods, and of course, developing some of the first relatively high throughput for their time systems for relatively deep analysis for its time with MudPit, analyzing over 1,000 proteins of the yeast proteome in a few hours of time or something like that. There was a time about 20 years ago when this was a massive feat, a real big improvement over what anything that had been done before. And all of these things took a lot of innovation. They took new ideas. And I think that's what our field of single-cell proteomics needs. We are now in the position where bulk proteomics was 20, 30 years ago. And we can take advantage of all of the creative solutions that were invented by leaders in the field, as John Yates for bulk proteomics. They can also work for single-cell analysis, but some of them may not be optimal. We may have to rethink how we are doing things and invent solutions that work better for single-cell protein analysis. And I expect that John will have a lot to offer with that. So I'm very excited to hear his presentation today. Just lastly, I'll say that currently, as many of you know, he is a professor at Scripps Research Institute. Welcome, John. We look forward to your presentation.
Thank you. Thank you for the very kind introduction. And okay, can you hear me?
Yes.
And hopefully you can see the screen. Thank you, Nikolai, for the kind words and the introduction and for the invitation to talk again. The title is a little bit different, but it's basically the same title. It's in the abstract program, in the program. This is really a collaboration between my lab, so Jolene Dietrich, and then members of Stuart Lipton's lab. And then new to this is people from Marissa Roberto's lab, Larry Rodriguez. So we've got two labs that are doing the patch clamping and working with the neurons, and we're doing the sample preparation and mass spectrometry on them. And so I would like to throw a couple of other possible names for the SCP2021. So Slavov Palooza 2021 or SlavovCon 2021, in addition to Slavov Fest, which I think we saw in the last talk. Okay, so just by way of introduction, so we're looking at neurons, and neurons are probably not the hardest for single cell analysis based on the size of the cells. These are very large cells, and for a little bit of perspective, I put HeLa size, although these aren't quite to scale. But neurons, what makes neurons tricky is that they have all these projections, and they're going out sometimes for very long distances, and they're forming connections with other neurons. And so the isolation of the neurons is actually what makes these things more difficult than it is the absolute mass of proteins that's present. These are actually fairly from the scale that people have been talking about at this conference, these are actually fairly large, but they are fairly sophisticated cell types, highly differentiated, and perform relatively important functions. And in fact, these things neurons form circuits that are capable of creating learning, memory, behavior, thought, and critical thinking. And so these are all the very sorts of things that make us human beings and allow us to do the kind of things that we're doing. And so it's understanding how that happens that has become a really kind of interesting and important to know. And a bigger brain is, of course, important, although we probably don't have the biggest brain of all the mammals that are out there, but more neurons with more synapses are important as well. And if you look at the scale of total neurons in the nervous system versus total synapses, you can see that Homo sapiens have quite a large number. And here is by example, is a cat. And so you're all probably familiar with behaviors of cats and the capabilities of cats and the intelligence of cats. And so they're about 10 to the 12, 10 to the 13 total synapses, where we're about an order of magnitude or two more. And of course, if you've seen cute cat videos on the web, you know that cats are actually quite smart. Cats sit at home and lounge around while their owners go off to work and do things. And so in order to cram more surface area within a small cavity, human brains have increased the number of folds that are in the brains as a way to increase the surface area and increase the number of synapses and neurons that are able to get in there. And if you've read, I remember reading Carl Sagan book many years ago, and that the ultimate limit of the human brain is going to be its ability for the ability of females to birth offspring. They can only fit the brain of so big out the pelvis. Let's see. Okay, so when humans die or our ancestors died, they left behind fossils in the form of bones and things like that. But what doesn't, the soft tissues don't leave anything, so they don't really tell us. We're not able to learn anything about the structure of the brain as it developed across the years. But one of the things that was left behind was artwork, and so this gave us an ability to look at the intellectual capacity of humans and pre-humans as they sort of evolved through time. And you can see this piece of art 300,000, 400,000 years, 500,000 years ago. You can see it's got some sophistication to it. It's starting to looks a bit like a human. And then 25 to 35,000 years ago, you see a little bit more. And then same thing with this Venus of Willendorf. And what humans are clearly trying to do in these instances is to recapitulate what they can see. And Michelangelo clearly does that with David, with some quite striking elegance in recreating the magnificent structure, magnificent physique of David. And then many years later, 600 years later, 500 years later, you see Jackson Pollock is not simply recreating what he can visualize, but he's visualizing something completely different with art abstraction. One of the things that I really like is building things and the capacity of humans to use their intellectual capacity to build amazing structures and things like that, such as Notre Dame, this compact muon solenoid, which is probably one of the most complicated things humans has ever built. But what also is intriguing is, if you've ever flown on one of these A380s, it's amazing that this thing gets off the ground, and it is important to understand that many humans had to have their synapses properly functioning for this thing to get designed, built, and to be flown without crashing into the ground. And we other humans trust that all those systems worked properly in order to build that plane and for that plane to get off the ground. It's a lot of trust in there, and trust in the capabilities of human beings. Okay, so the primary challenge with human brain studies is it's difficult to study live human brains at the molecular level in situ. Most humans, there are many organs and tissues in the human body that you can biopsy and you can sample. It's not so easy to do that with human brains unless somebody's being treated for some sort of disease like a brain tumor or something like that, where you can go in and get some tissue. But ordinarily, people don't let you mess around with their brains. So most studies of live human brains use non-invasive spectroscopic methods such as functional MRI. More recently, in the last 15 years or so, 10, 15 years, optogenetics has come along, which has been really quite a remarkable tool to allow interrogation of brain circuitry. And what we've learned from these types of studies is that there are discrete circuits that are involved in regulating certain kinds of behavior. More recently, human brain organoids have come along, these mini brains, which allow the study of disease genotypes and possibly function. It's just not clear how well functions will be recapitulated in organoids. It's a 3D organization of neurons that form circuits to carry out function. Now, one of my collaborators, Alison Mottry, has showed that brain organoids have similar electrical activity to fetal brains. So you see very similar sorts of electrical patterns in organoids that you would see in the fetal brain. And it wasn't quite an apples to apples comparison, the study that he did, but it was about as close as you can get without, about as close as you can get with when you're working with fetuses. So single cell molecular analysis in neurons is a very long history. A lot of it's been led by John Sweedler's laboratory and others in neurons, such as Aplasia californica. And Aplasia californica has a very important place in neurobiology. It was used by Eric Kandel to try to understand memory formation. And he got a Nobel Prize for that at one point in time. And it's still considered to be one of the premier model systems to be able to study neuron activity, neuron chemistry. But as Sweedler's lab developed these techniques and improved on these techniques, they started to apply these to other types of cells. And they used a variety of techniques such as MALDI, CE, LC, MS, and measured a variety of different molecules. There's not been a lot of progress on proteome analysis in single neurons. Mostly it's been neurotransmitters and neuropeptides. But an important point is that Sweedler's lab and a lot of his offspring have helped establish the importance of single neuron analysis. And these are just some of the papers where measuring peptides in individual organelles with mass spectrometry, almost done 21 years ago. And looking at feeding circuit activating peptides, neural peptides from Aplasia. And again, this is just slightly under 20 years ago, where Sweedler's lab has looked at these single cells and measured important molecules, discovered important molecules with these systems. And one of his offspring, Jung Lee, who I noticed is on this at this meeting, has also continued a lot of this work looking at single neuron analysis in crustaceans, but using techniques like imaging analysis and so forth as a way to get at that information. Okay, so a couple different ways you can look at single cell proteomics in neurons, and we're doing both. So one is to use iPSC generated neurons. It's good to study the effects of genotypes on electrophysiology and connect to molecular profiles. And you can generate these neurons, and you can do 1D cultures and 2D cultures, and now you can do 3D cultures of the formation of organoids. And that allows you to look at more complicated systems, how well the 3D cultures and organoids recapitulate normal brain activity is still up for debate, although people are starting to use these with some pretty good results to screen for drugs for certain types of neuropsychosis and so forth. So brain slices you can combine with learning protocols to study behavioral effects on neuronal circuits within brain regions. You can look at animal disease models and so forth, and tissues are more complicated with additional cells and extracellular matrix that you have to contend with when you're trying to look at individual cells. And so we started by looking at iPSC generated neurons to combine electrophysiology with single cell analysis. And we're now starting to moving into brain slices as a way to do this. And this has come with some additional sample handling issues that we have to deal with. So one of the first ones we started with was Alzheimer's disease. This is a collaboration with Stuart Lipton's laboratory. And this literally started as a hallway conversation talking about how we are going to start moving into single cell analysis and and Stuart had some projects he wanted to work on with this. And so Alzheimer's disease, you all know about, it's about 5.8 million Americans, 50 million worldwide, 10 million new cases each year. Particularly acute problem in the United States as the baby boomers start to age, and this results in very high societal cost. And these result in problems with memory loss, problems with language, disorientation, mood swings, loss of motivation, and so forth. So there are lots of hypotheses that do with Alzheimer's disease and what causes it. The A-beta hypothesis has its biggest test now, that there's now a drug that's been approved to clear the amyloid-based plaques that are formed, and we shall see if that is useful or not. But it's clear that there's a massive failure in proteostasis involved, and the genes involved in familial AD are well understood, but sporadic AD is still a bit of a mystery. And these are sporadic AD is by far the most cases that are observed. We've certainly gotten some great clues from the familial AD. So one of the hypotheses that's out there is one of the A-beta hypothesis proposes that these plaques that are formed by the A-beta dimers and oligomers lead to neurotoxicity and neuronal death. Another hypothesis along that same line is that not that the amyloid plaques are in fact neurotoxic, it's that they lead to hyperactivation and eventually neurodegeneration. So the cells are triggered and they're very hyperactive, and eventually they just kind of wear out, I guess. So that's one of the questions that we're following up with the Lipton laboratories to try to understand this hyperactivation and what are the molecular changes that are going on inside the nucleus, inside the neuron. And so we are generating, or Lipton's lab is generating human-induced pluripotent stem cells. They take wild-type neurons from the skin biopsy of an 18-year-old male, a normal, healthy 18-year-old male. And then these AD neurons are generated by inducing a methionine 146 valine mutation in the presenilin 1 gene of the wild-type PSCs using CRISPR-Cas9. And these primary, the skin biopsy is the primary fibroblasts from the skin biopsy are then activated with Yamanaka factors. These reprogram to the embryonic state. We then get iPSC cells, and then they're differentiated the neurons by dual SMAD inhibition protocol to form NPCs, neuroprogenitor cells. And these are subsequently treated with BNF and GDNF. Those are neurotropic factors. And then they're plated with mouse astrocytes and they undergo neuronal maturation. So after about 4 weeks, you get mature neurons, which are ready for experiments. And you might note that there are two of the methods in here are Nobel Prize methods. So Duden and Charpentier for the CRISPR-Cas9, and then Yamanaka for the iPSC cells. And then the cells are then analyzed with a third method for which a Nobel Prize was awarded in 1991, which is this patch clamp technique. And so there are probably fundamentally two approaches to doing single cell analysis. One is that you're looking at a lot of different cells and you're trying to collect a lot of these different cells and try to make sense biologically of all the different things that you're able to glean from the large numbers of cells. And the other strategy would be to do extensive phenotypic characterization on a single cell, and then analyze that cell and try to connect the phenotypic information that you're able to derive from the cell with the molecular information that you can get from the molecular analysis of the cell. That's what I look at. That's how I look at what we're trying to do here with by combining electrophysiology with the proteomics. And so what you can see here is the pipette tip, and it's interrogating a neuron in culture. This is a 2D culture, and so you can see that you're able to put the tip on a cell and then measure it. And there's lots of interesting information that you can glean from this. This is a wild type neuron here, and this is whole cell patch recordings And this is the AD neuron over here, which you can clearly see that this cell is hyperactive. And so these are the spontaneous action potentials. And so neurons are just randomly firing, and with sodium channels open up and they allow sodium to pass out, and that creates a electrical pulse down the neuron. And when the cells are hyperactive, you see lots and lots of firing. And so what you can see here is that the wild type cell doesn't fire as much. The AD cell is firing quite frequently. And there's also you can measure capacitance of the neuron, and that tells you something about the cell size. And so in the wild type cell, what you can see is that the capacitance is quite high. That suggests that the cell is bigger, it has more dendrites. And on the AD related cell, you can see on the AD cell, what you can see is that there are it's smaller, the capacitance is lower, and so there aren't as many dendrites. And if you look at do some staining, so you can stain for MAP2, microtubule associated protein 2. It's a marker for dendrites. And so that's the red color that you can see here in the wild type. You can see there's far more of it. Then if you look at TUJ1, which is a class 3 beta tubulin, it's a pan-neuronal marker, and you can see its color is green, and you can see that there are many more. There are also a lot of neurons here, pan-neuronal markers on this one. And then when you look at the levels of it, you can see that there's far fewer dendrites that are being formed on the AD neuron, and there aren't as many neurons in the AD neuron as well when you look at the pan-neuronal marker. Okay, so these are the kinds of things that you can measure when you do these patch clamp protocols. Measure action potentials. You can measure sodium current density, capacitance, spontaneous action potential frequency, as I talked about earlier, potassium current density, and resting membrane potential. And so you can record all of this data on the membrane. You can record it for inhibitory neurons as well as excitatory neurons. And this is data that is shown for the 20 AD neurons that we characterized by patch clamp, as well as the 24 wild type neurons that were also patch clamped. And so these all passed the screening protocols and were collected for mass spectrometry analysis. And the electrophysiology conformed with what we were expecting for a wild type neuron versus a AD based neuron based on what's been reported in the literature previously. This is the protocol that we used. So we did the whole cell patch clamping. The patch clamp was put down, and the patch clamp is really just a nanospray like needle from the 1990s. And in fact, the devices used to pull those needles were first used in patch clamping experiments. Sutter Instruments sells a lot of these things, and they're primarily used in patch clamping labs. So after we do all the measurements, it takes about 20 minutes to do the measurements. We then apply some slight negative pressure on the patch clamp, and that allows us to withdraw the neuron from the cell culture. It's then expelled into an Eppendorf tube. We then treat with trypsin, and it's incubated at 60 degrees for about an hour. We digest it up, and then these first sets of experiments were run with a 140-minute gradient with an easy nano LC and on a thermo eclipse. We collected data for about 44 different cells using this protocol. It didn't take much effort to get some data out of these systems. What we did mostly is we shortened up the incubation period with trypsin and elevated the temperature, and that allowed us to very quickly get data out of this system. It didn't take a lot of effort to get to the point where we were starting to see some data. And this is the proteomics pipeline that we use. So the Orbitrap Eclipse mass spectrometer, and then the data analysis was done with our internal database search engine, ProLucid. We used the UniProt human database, and then the search results were filtered and compared with using Pattern Lab. And this was a program developed by Paulo Carvalho. It's got some nice capabilities for doing NSAF for comparisons, as well as some nice plotting tools for gene ontology and so forth. And so we use that. And it's also a Windows-based program, so it makes it a little bit easier to use. So single cell analysis, you can see here that we have a range of proteins that were identified from the AD versus the wild type, almost up to about 1,500 proteins, in some cases down to only a few hundred proteins. So we, out of the 44 runs that were done, one failed a mass spectrometry run. Peptides identified ranged from thousands to almost over 6,000, 7,000 peptides identified. So if we filter the data, so we wind up with data for about 14 different AD and 18 different wild type cells. That's about a 70% success rate. We get approximately 700 proteins on average, 2,000 peptides identified per single cell. But you can clearly see that there's a range of peptides identified, as well as a range of proteins that are being identified in these different experiments. And as best as we can tell right now, that is related to our sampling handling strategies. So it's the extraction of the neuron out of the culture. It seems to be the biggest variable at this particular point in time, and we need to improve on that. So here's Venn diagrams just showing identification in a minimum of 3 samples per genotype. So these are the proteins that are present in at least 3 cells per genotype. And if we just look at proteins that are identified in 1 sample per phenotype, we see that there's a fairly big difference between the wild type and the AD. And then if you plot differential protein expression in wild type versus AD, my buttons aren't working. One of the more interesting things is the immunohistochemistry marker that's used for dendrites. You can clearly see that it corresponds to what you would expect based on the imaging data that we had generated for this. But there are about 32 proteins. And 42 other proteins that satisfy fold change and significant p-value. So yellow are low abundant proteins and the blue are high abundant proteins. So we are clearly seeing some changes based on the data that we've obtained so far. And this is differential expression using a heat map to show these differences. You can't read this. I can't even read this from here. It's a little bit blurred. But you can see that we can plot out. We can see differences among the AD versus the wild type with this particular data that we got. And this is another plot showing the partial least squares loading plot of the AD versus the wild type samples. So clustering analysis. We also did some reactome pathway analysis, which shows coverage of the overall protein identifications. And we see that we get some data from most of the reactome pathways. And that's not to say that we are covering high levels of the pathways, but we're getting at least 1 or 2 proteins into the pathways that are observed here. And if you also look at the significantly changing proteins, the ones that are in yellow are ones that are being matched up to some of these pathways from a reactome. And string analysis from significantly changing proteins. This gives us some interesting information. If you look at the cellular component from gene ontology, proteasome, which you might expect to be upregulated. Given that this is Alzheimer's is a proteostasis problem, but also basal dendrite, astrocyte, end foot, cytoplasmic dynein complex, and the dynein complex as well. Where'd it go? Okay, so we've also been started looking at brain slices. This is a much harder problem for a variety of reasons. And my neighbor across the hall, Marissa Roberto, is an electrophysiologist. She also does alcohol addiction studies. We started working on this when a a faculty member at Scripps died suddenly, and his postdoc needed a lab to go to in order to finish up his work. And so he came into my lab, and he was working on some alcohol addiction studies. And so this got me involved, got me to know Marissa, and we started working together. So alcohol addiction in Scripps has a very long history. There's a neuroscientist you may have heard of or not, Floyd Bloom. He was at one time was the editor of Science magazine, and he 35 years ago he approached the NIAAA and he asked if they would be willing to fund an alcohol addiction research center under the idea that addiction may in fact be a biochemistry problem and not a problem of hedonism. Up to that point, everybody thought that addiction was a character flaw and not a biochemistry, neurobiology issue. And so they funded his center, and they funded a bunch of other centers, and now there are a lot of alcohol addiction centers that are in the United States that are studying alcohol use disorder. One of the model systems they used are rats. Like people, rats will self-administer alcohol. Mice do not. Mice are smarter than rats, but rats will self-administer alcohol, and so they can become addicted to alcohol quite easily. And in fact, you can see that these rats are drinking some wine, and I think they must have taken it from my collection of wine in my office. And you might think that I drink a lot, but this has actually been accumulating for the last 25 years. And that little green box back there is a bottle of 1998 Dom Perignon that was given to me when I got tenure by someone. You can see it's still back there. And there's some Maltai there, which was given to me by Chinese scientists, still in the box. But we can, the rats can be addicted, but to alcohol, and we can then look at them. The central way that Marissa has been studying this for a long time is they do 1 millimeter punches from the central nucleus of the amygdala. You do then single pot 1 hour digestion, and then we've been using the EvoSep and the timsTOF Pro and the Pacer search algorithm, which searches in real time while the samples are being run. We're able to get about 15 samples a day. Now we're using about an 88-minute gradient. We're using the 88-minute gradient on this. So the central amygdala, so this is an area of the brain. You can see it's all mapped out here. So it's this little area of the brain here. It's implicated in behaviors related to fear, stress, anxiety, and alcohol dependence. And so this the connection between the alcohol dependence and the anxiety and stress may be why when you are feeling stressed or anxious, you might have a drink and then calm down a bit. So it's a way to self-medicate, but it seems to be the region that's implicated in alcohol addiction as well. It's predominantly GABAergic neurons, both projection interneurons. The central amygdala is the most peptide-rich region of the brain. And the GABAergic transmission plays an important role in mediating alcohol and stress-related behaviors. And so you can see where they will go in and they'll take these punches right here. And we did some proteomics on these. After she targets these sites by doing electrophysiology, and then she can tell which of the neurons that are responding in a way that she would expect given the alcohol addiction or the control, and then we'll harvest that region. And then we ran these for her just to get some background data before we started doing the single cell analysis. And what we found is that so we did male controls, male dependent, male withdrawn, and we found pretty good data by running this. We've seen about 3,000 proteins or so per experiment doing these experiments. And the 1 millimeter tissue punches is not a whole lot of material that's being used. So I won't talk too much about the software, but we have some software which has the unfortunate name of PCETSA, which kind of overlaps with the thermal profiling name strategy, but it's a way for us to look at proteomic data that comes from a mixture of bulk cell experiments and be able to assign proteins that are coming from specific cell types within that mixture of a tissue mixture. And you can see it here, and it's showing pretty good results. Shows that our tissue punches are primarily neuron, although there is a fair amount of oligodendrite, that dendrocyte that's present in the cell punches. We're still trying to vet this software to make sure that it actually works. Gene ontology, differentially regulated proteins are shown here. You can see these. And then this is from Larry Rodriguez. So if you think running mass spectrometers is art, you should try doing electrophysiology, where you have to patch clamp onto a single neuron and then apply suction, carefully remove the cell from the tissue, retract the electrode, break the tip into a C18 trap column, and then inject neuron and recording solution into the trap column. And then we go from there to do the proteomics. And again, the proteomics is pretty much the same as I've already talked about. Once we digest the trypsin, and then there's an 88-minute gradient on an EvoSep with a timsTOF Pro. And this is just some of the first data that we got off this. And you shouldn't that the numbers actually look pretty good, particularly the withdrawal. But this is not what you might expect. Is that the control brain with no alcohol is nice and calm, only 1,482 proteins being expressed. And then dependent brain is getting a little more agitated, 2,000 proteins, and then withdrawal, the brain is screaming, give me more alcohol, and it's overexpressing an additional 1,000 proteins. But what this really represents is a progression in Larry's skills in recovering neurons from the brain slices. And what's encouraging, which is why I'm showing this, which is encouraging, is that we're able to see about 3,000 proteins from a single neuron out of the brain using this strategy. So this is, as Andrew Leduc said yesterday, this is single cell analysis is really all about sample handling, and we are still working on our skill set to improve on this, so we can be not only good at the sample handling but also consistent with the sample handling. And this is a Venn diagram showing that the dependent versus the naive versus the withdrawal protein must occur in 2 or more samples. And you can see that there are fairly different numbers. Okay, so conclusions and further work: successful implementation of patch clamp, and we've successfully implemented this on single neurons. It's while time-consuming and low throughput, the patch clamp screening for neurons prior to selection for MS analysis is essential to obtaining high-quality neurons that electrophysiologically behave as expected. Some options to increase our throughput would be to implement the Slavov TMT method. That'll require some innovative sample manipulation protocols, but I think on the first day I saw a potential strategy for how we could do this. We'll have to take a look at it because the neurons are bigger cells, and so that may require some modifications. Minimal manipulation and sample handling was key in providing high quality MS data. We still need to improve our consistency in sample collection. For iPSC, we identify between 700 and 2,000 proteins per single cell, and most of that variation is related to sample handling. We analyzed 20 to 24 single cells per genotype. We're able to detect differentially regulated proteins, and many of which have been shown to be modulated similarly in the AD literature. We've observed several differentially regulated proteins in the single cell MS data that are highly correlated to decreased neurite length, and that we observe in our immunohistochemistry data, and this could lead to lower capacitance values. Current data obtains measurement from both excitatory and inhibitory neurons, and further categorization of proteomics data based on electrophysiology parameters could lead to additional information. So a lot of this was just preliminary data that we were collecting in order for grants, to get grants, and we should have a grant funded. If a fourth percentile doesn't get funded, then I'm in deep doo-doo. So we'll have a positions available for single cell proteomics to advance these methods, and will be to study iPSC and organoids that are related to autism, study autism. So people that have done all this work, so Jolene has done a lot of the sample handling on our end to do the mass spectrometry and the proteomics. Some of the others that have helped with Salvador's done a lot of informatics. Stuart Lipton's lab has done the iPSC cells for the AD study, with a lot of it being driven by Matt, but Swagata and Nima have done the the patch clamping. Larry Rodriguez has done the patch clamping, and Marissa Roberto's lab, which is done on the alcohol addiction studies. And some of the work's been funded by the National Institute of Health, and some of it was for preliminary data to try to get some more funding. And thank you very much.
Thank you, John. That was a treat. We have a few questions from the virtual audience that I'm going to start with, and then see if we have any questions from the in-person audience. From the virtual audience, we have a question for AD versus wild-type neurons. Did you look into post-translational modification changes in specific proteins? For example, phosphorylation on tau.
Yeah, not yet. This is still all very preliminary data. I'm not even sure if we're seeing tau in there. I'd have to go look. But yeah, ultimately, that's what we would like to be able to do. We'd like to be able to get enough sequence coverage that we can see modifications. So in a number of the previous talks, we had discussions about, there were discussions about being able to see PTMs and so forth. So one of the tricky things that single cell analysis is going to be trying to do this without enriching, because that's going to add another step to the process. And every time you add a step, you're going to suffer for losses. And so I think our end goal really should be to improve our sequence coverage dramatically, so that we can have a better chance of seeing modifications.
Thank you. Another question is from Jennifer Heck. She's saying, great combination of phenotypic readouts and molecular underpinnings. You have briefly mentioned the electrophysiological recordings of inhibitory and excitatory neurons. Do you induce a pure
glutamatergic
culture system using the differentiation protocol you mentioned? Have you compared the effects of the AD mutation on induced glutamatergic versus GABAergic neurons?
Not yet. A lot of that's being driven by the Lipton laboratory. They're also collecting RNA-seq data on this. And right now, so we've collected a lot more data on these systems. And right now we're trying to figure out how to put all of it together. It's so that we can make some sense out of it. So yeah, there's still a lot more to do, but at the moment we're flying without a net because we don't have any actual funding for this project.
Well, it's good that you did receive some funding. It seems like it, and it sounds like an exciting opportunity for students who would like to do a postdoc in excellent lab. So we have another question, starting with great talk, John. Any thoughts on trying stimulation of neurons of protein expression, maybe through the use of pulsed Silac?
Yes, I haven't thought about pulsed Silac, but what I have been thinking about, so we've been doing a lot of work with azidohoma-alanine. And so we have been thinking about how to, I've been thinking about how to tie that in to this. And so we can look at new protein synthesis as a function of some perturbation or some conditional stimulation. In particular, so I've got a lot of the PIs in the alcohol, our resource center, research center, thinking about using bioorthogonal chemistry, so BondCat, as a way to look at what changes are going on when the animals start drinking alcohol. And there is a new a strategy, cell-specific, not that new, but it's reasonably new. It's cell-specific BondCat that's available using the MetaRS mouse, and we're doing experiments with that and so forth. But yeah, we'd love to be able to do something like that.
Thank you. Are there questions from the audience here? I have a question, maybe more of a suggestion, but I think it's very exciting that you're able to collect functional data about neuron firing and potentially correlate this with molecular abundances of various ion transporters on the plasma membrane. So have you tried to just correlate the protein abundance of various ion transporters to the electrophysiological recordings that you have?
Yeah, that's clearly an end goal. The business end of these neurons are the synapses, and at those ends there are tons or lots of receptors and ion channels, which are going to be incredibly important to this function, and those are end goals for us to get to.
Well, they're great end goals. I look forward to see what the results are going to be. Oh, I see there's just another question popping out from Brian Fonslow. Let me quickly ask.
But there's a one before that. Rawad?
Yes. So nice talk. Just out of curiosity, is it possible to mimic alcohol addiction in a tissue culture dish, or is it too complicated?
So that's an interesting question. So one of the things that these iPSC systems allow us to do is to look at the genotypes and how that affects the phenotype. In particular, how it might be affecting protein expression and so forth. And it is known that alcohol addiction runs in families. It seems to be genetic in its transmission. And so one of the questions that's come up in our meetings is whether or not it would make sense to get skin biopsies from families where alcohol addiction runs through the family, and to make tissue culture systems and to try to look more closely at the molecular perturbations that might be going on in these systems. I don't think you can feed the cells alcohol and see the same kind of effect, but you might be able to look at protein expression and see what sorts of systems are different.
Cool. And the last question is starting with great talk, John. The patch clumping measurement and the needle isolations of neurons is cool. It seemed notable that immune processes and protein turnover had protein abundance changes. Similar to the post-silo question, are there other strategies to specifically target and understand those?
Yeah, so there are some strategies to look at protein turnover. Again, the post-SILAC would be one. We've looked at protein turnover using BondCat, the azidohomoalanine system. But some of these, not the SILAC per se, but certainly the BondCat, require some additional steps. And I've always defined protein biochemistry as being like a chess game, where you want to win in a minimum number of steps. Otherwise, the more steps it takes, the more likely you are to lose or to end up in stalemate. So anytime you have to do additional enrichments, particularly with these small amounts of material, you're probably going to lose too many things. So post-SILAC is probably an easier one because you're not going to have to undergo an enrichment method. But I think the ones that we like, the AHA, might be out of the question for a little bit.
Great. Thank you.